Emil Fischer won the Nobel Prize in Chemistry in 1902. But wait, what exactly did he accomplish to earn this mighty prize? A molecule called purine is present in your DNA, uric acid, coffee, and tea. This molecule is a vital building block of life. Emil Fischer cracked its chemical structure and successfully recreated it in the laboratory. This was the first time a complex organic molecule of this nature had ever been synthesized. Doing so, he laid the foundations of modern genetics. He continued his work and became the first person to determine the exact structures of sugars like glucose and fructose, synthesizing them in the lab as well. He then unlocked the secret of how enzymes break down sugars in our bodies, earning his place as one of the founding fathers of biochemistry. He went on to synthesize amino acids and octadecaptides, proving that proteins are polypeptides and laying the foundations of protein chemistry. A genius far ahead of his time, Emil Fischer was awarded the Nobel Prize for his matchless contributions to science. Let's take a deep dive into the life and work of Emil Fischer. Hermann Emil Fischer was born on October 9, 1852, in the small German town of Euskirchen. His father wanted him to run the family lumber business, but Emil wanted more. At school, Fischer struggled with languages, but had a strange gift. He could see numbers and shapes in his head. Molecules weren't formulas to him, they were puzzles. By the age of 20, he'd left the family business behind and enrolled at the University of Bonn. It was here that Fischer met a mind that would shape his destiny, Adolf von Baeyer, the pioneer who later won the Nobel Prize himself. In 1875, Fischer's world changed because of what seemed like an accident. While working with a strange chemical called phenylhydrazine, he stumbled upon a tool that would unlock nature's secrets. With this, Fischer cracked open the architecture of sugars, glucose, fructose, mannose, shaping the Fischer projection, a simple diagram that chemists still use today to visualize molecules. Imagine a man in the 19th century sketching sugar's structure with a pencil, and more than a century later, every biology student in the world still studies those same drawings.
By the 1880s, Fischer wasn't just studying sugars, he was decoding purines, the molecules hiding in your morning coffee, your evening tea, and even inside your DNA. Caffeine, theobromine, uric acid, adenine, guanine. Fischer mapped them all. Without his discoveries, we wouldn't understand the double helix. We wouldn't understand genetics. We wouldn't even understand why a single sip of espresso wakes you up. Imagine billions of tiny machines inside you right now, tirelessly working, never resting. These machines are enzymes, molecules that make life possible by accelerating every chemical reaction in your body. In the late 19th century, no one understood how enzymes worked. Scientists were puzzled. How could a molecule choose its partners with such perfect precision? Then came Emil Fischer's revelation, one of the most elegant ideas in biology. Enzymes and molecules fit together like a lock and its key. He proposed that every enzyme has a uniquely shaped pocket. Only the right molecule, the key, can fit perfectly into that lock. One wrong shape, and the reaction simply won't happen. This model explained why your body knows exactly how to break down sugar but ignores toxins, why your cells can distinguish glucose from fructose, why life itself has order instead of chaos. Think about it. Without this mechanism, your blood wouldn't clot, your food wouldn't digest, and your DNA couldn't replicate. You simply wouldn't exist. What's even more astonishing, Fischer proposed this idea decades before we could even see enzymes under a microscope. He saw nature's invisible dance through sheer reasoning, curiosity, and imagination. Today, entire industries, from pharmaceuticals to biotechnology, still rely on Fischer's lock and key principle. Every life-saving drug you take, every vaccine you receive, every cutting-edge cancer therapy being designed, all of it echoes Fischer's vision.
In 1902, Emil Fischer won the Nobel Prize in Chemistry for his groundbreaking work on sugars and purines. He became a global icon, earning honorary doctorates across Europe, joining elite scientific academies, and shaping generations of chemists. But while the world celebrated his genius, Fischer's personal life was falling apart. Fischer married Agnes Gerlach in 1888. She died just 7 years later, leaving him with 3 young sons. Then came the First World War. One son died in combat, another took his own life. The man who had unlocked the language of life was surrounded by death. His health crumbled under years of chemical exposure, his heart broken beyond repair. By 1919, Fischer's body was failing. After decades of inhaling dangerous chemicals and living through unbearable grief, he faced terminal illness. On July 15th, Emil Fischer took potassium cyanide and was gone. But his work, his fingerprints, remain everywhere. From insulin research to genetic engineering, from drug design to everyday medicine, Fischer's discoveries are part of our lives, quietly, invisibly, powerfully. He didn't just map molecules, he mapped life itself. Next time you sip your coffee, think of the man who decoded its secrets. Do you believe that one person today could change biology as deeply as Fischer did back then? Tell us in the comments. This is Awards, where the world's greatest minds live forever. Don't just watch history, be inspired by it.
Thanks for being with Awards. Subscribe to know more about international award winners and unsung heroes behind peace, science, and humanity.